Well, thank you very much. That's quite an introduction. Thank you very much for staying here. First of all, I admire your tenacity and doing all this. Caffeine is right out there somewhere, so I suggest I wouldn't feel insulted at all if during my talk you go outside and have some coffee. I was asked to talk about the metaphor of mitochondria. That's the title Professor Cittadini gave me. And I thank him very much for that title. I'm still struggling with it to see what it really means. It's, of course, an abstract, poetic version of what my talk should entail. And there are so many aspects of mitochondria that to condense it all in about 20 or 25 minutes is not that easy. So let me get on with it and see how far we can get. So here are my potential conflicts of interest. I think that you'll see from the content of this presentation that most of that doesn't apply to conflicts. So the outline is, first of all, for those of you in the audience who are new to this topic, I'll go very quickly over mitochondria, mitochondrial mutations, mitochondrial disease, copy number, talk a little bit about disease treatment options, and there are quite a few now available, all potential, all experimental, PGD for mitochondrial disease, which is the one I prefer. Mitochondrial replacement therapy, which is obviously the one that is famous, particularly among the popular press, because it's a spectacular way of dealing with this problem. And it's also a way that has been popularized, particularly by advance in the political arena in Britain, where this thing has been debated openly, which is an astonishing thing. It's not debated at all in my country, in the USA, where this basically is prohibited. Then there's the option of mitochondrial DNA editing, which Christina already referred to, not in the context of mitochondrial DNA gene editing, but you could do that, and it has been done in the mouse. Talk about it very, very briefly. Then mitochondrial counting, mitochondrial copy counting. There are so many mitochondria in eggs and embryos that could potentially be correlated to outcome. And of course, there have been about a dozen papers or so on this topic in the last 3-4 years by Elpida Fraguli and Dagan Wells and others. And so I will not really cover that because that's kind of a topic on its own, although I'll hint at it maybe sometimes in my presentation. Then cytoplasmic changes by mitochondrial replacement therapy or mitochondria transfer. That has been done for the last 20 years with different results, very poor publications or no publications at all, in repeated implantation failure or in patients with advanced maternal age. I'll cover that also a little bit if I get to it at the end, but feel free to interrupt me if I go over time, because I think this session has gone on long enough, and you still have 1 or 2 speakers to go. So this is, of course, is a transmission electron microscopy graph picture of an adult mitochondrion and an adult somatic cell, and you can see a lot of membranes. The little black dots are small mitochondrial DNA loops, and in mitochondria of adult cells, like in muscle cells and brain cells, you'll see up to 10 or so of these copies. These are copies of single-stranded DNA, a small number of base pairs, 16,600 per, about 16,600 per mitochondrial loop. It's circular, single-stranded, and the DNA sequence has been determined in the 70s, so it's been known for quite a while. Association with mitochondrial disease also dates from the late 70s and early 80s, actually mid-80s, where these rare diseases were associated with mutations in mitochondrial DNA. Now, the egg mitochondria and the mitochondria in the embryo don't look like this at all. And there are similarities, like the double membranes that you see, a lot of membrane structures also inside the cells, but they all have only one copy of mitochondrial DNA. At least that's what we are being taught. Whether that always applies to every mitochondrion is probably not likely. There are probably mitochondria that have more than one copy, but they are very, very rare, at least in microscopy. And so they're small, they're round, and kind of uniform like this. So they're very different from somatic copies of DNA, mitochondria. This is a cartoon of the mitochondrial DNA, and consists of 37 genes. Most of them are involved in transfer RNAs, some in ribosomal RNAs, and then there are 13 protein coding regions. Not all of the genes involved in OX-phos metabolism and energy metabolism are located on the mitochondrial DNA. The majority, the vast majority, is actually on the nuclear genome. So PGD for mitochondrial disease transmission, or avoiding mitochondrial disease transmission, has been done for many years already on patients who had the affected gene on the nuclear genome. It has not really been done until about 10, 12 years ago. It started, I think, in France, in René Friedman's group, where the occasional patient was done, and these were all documented and published. It started, I think, in 2006. There is a little area on the top here in this cartoon at 12 o'clock. It's called the D loop. It's a hypervariable area. It doesn't code for a gene. However, it's very important because it's involved in the replication process. And this is usually where there is hypervariability, meaning that this is where you can sequence an individual and have an individual fingerprint. So this has been used for the longest time in all areas of, many areas of medicine and also in forensics. So there are many mitochondria per cell. These are somatic cells. The ratio is 1 in X in early embryos, which makes it a bit easier to deal with them. But there are so many mitochondria in X and in embryos. In X, there's at least 50,000, and you have to look at the studies to sort of comprehend what the ratio is. It's probably very variable from egg to egg and from one cohort to the next. There are maximally, the counts that I've seen in the literature were 700,000. So this is enormous variation, but there are a lot of them. So how do you deal with that when you want to determine mutations, or when you want to deal, when you do with copy numbers? So it's not the easiest process. Replication of the mitochondria is not synchronous with meiosis. You can't really argue replication starts at any point. It's an ongoing process, although it stops also at some times and then starts again. I'll show you a copy of that. So how are mutations removed? Because it's a single-stranded haploid DNA. How do you deal with mutations? Because when you have a haploid DNA, you can get accumulation, almost like asexual reproduction, in terms of mutation load going to the next generation. So how that's dealt with in a way by something that's described by the bottleneck, which is there are probably several bottlenecks where these mutations are removed. And that's either by genetic drift or stochastically. Mitochondria are seen in all cells of the body except for red blood cells, and they deal with energy metabolism in another metabolic way. They're so short-lived that they can get away with this type of mechanism. But it's very different from other cells, which is interesting. This is an important little graph that I wanted to show you. This comes from Justin St. John, who's now in Australia. And in fact, if you follow Justin St. John's literature, if you look for his literature, you'll get a pretty good overview of reproduction in mitochondria. A lot of publication. Another one that, if you're more interested in the clinical line of this work, I would suggest you read papers by Bert Smeets from Maastricht in the Netherlands, and Joep Gerards, who's the next speaker, is co-authored on most of that work. And those two populations of scientists will give you a pretty good overview of the exciting work that's going on in this field. But anyhow, this is a log scale, and you can see here in the timeline, going from primordial germ cells over here all the way up to the mature oocyte, and then down to trophectoderm and ICM cells, that these copy numbers change a lot. So those of you who are interested in looking at copy number and whether you can do embryo selection based on it, the difficulty is that you can't compare one stage to the other. So comparing work that's done on the 4-cell stage or the 8-cell stage, you can't compare that to day 5 or day 6. And probably you can't compare day 5 and day 6. You probably have to correct for that somehow too. Or at least keep that in mind when you look at the literature. So it's a dynamic process. Okay, so mitochondrial disease. There are quite a few of them. There are ones that are known, that are listed here in the first line. If you look at the mitochondria that have mutated, it's not 100% or 0%. It's not like Mendelian genetics, okay? So it's a ratio effect, usually. In some cases, like Leigh syndrome, you can have very high levels of mutation loads. In some patients, that can be as high. In some embryos, it can be as high as 100%. And most of those will miscarry. But the mutation load in those that are affected can be between 20 and 80%. That's kind of what we aim for. And it again was the group in Maastricht who did it. They did a systematic review about 5 years ago, and they published that if you actually look at all the data, 18% mutation load is a good cutoff ratio to argue that you can apply that to almost any of these diseases and be in a safe range. If an embryo has 18% of a mutation that you're looking for, then you're probably below 18% or 18% included and lower, you're probably safe. Everything above it, you may have an embryo that is affected. Though, of course, may be exceptions to this, but that is about the thing to do. The advantage of that rule is that you can now apply it to any disease, not just look for a disease and say, or look for the patient and say, well, the patient has 25% or 28%, but the offspring of the patient was affected at 35 or 50 or 80%. Then you have to recalculate it for every patient, for every cohort of embryos that you look at. So rather than doing that, it may be better to just take one cutoff, and Bert Smeets and his co-workers set that at 18%. And I believe that is kind of accepted, and they have done a pretty large series recently that I think haven't passed, at least I haven't seen the publication, but they have presented in several meetings where they have quite a few pregnancies, and the pregnancy rate that to me looks quite normal for IVF. In a number of patients, it's somewhere between 60 and 80 patients that they have looked at. So there is progress. This was done with PGD, so there is progress in this area. So what you do is you have to look at heteroplasmy. So heteroplasmy is looking at this mutation load, anywhere from 0 to 100%. If it's 0 or 100%, those are then both called homoplasmy. If it's 100%, it's homoplasmy for the mutation. If it's 0%, it's homoplasmy for the wild type. Of course, we all have heteroplasmy. As we are sitting here and standing here, we all have heteroplasmy, and it becomes more severe with age. The disease is untreatable. It's not reversible. They're fortunately rare diseases, but they're very, very tough diseases to deal with. 15% of all the mutations are mitochondrial DNA, 85% is in the nuclear genome. The myopathies are the most common ones, but it is a subclass. The symptoms are neurological, learning disabilities, they're muscular, visual, digestive tract related, kidney related. Often there are multiple systems involved. And the cause is, of course, a depletion of ATP, but also there may be the generation of reactive oxygen, which may interfere with metabolic processes. It's very person-specific. Almost every patient is in their own group. So what are our options in embryology? And these are all exciting options that started in 2006 or 2005 in Paris, doing PGD with PCR-derived technology. That's one option. So you take the ones that have a high mutation load out, and you only transfer the embryos that have a low mutation rate. You can do mitochondrial replacement therapy, or you can do genome editing, or you can maybe do combinations of these. Like one of the debates is if you do mitochondrial replacement, and you do this intense nuclear transplantation technology, you probably should still do some PGD afterwards. And it has been debated in Britain, and I think that those who argued against it won that debate, which is interesting. But you could certainly do PGD and then combine it, maybe in the future, not anytime near, with gene editing. So PGD, or PGT as it's called, there were less than 50 cases when I put this together from the published literature. There are less than 50 cases of PGD for mitochondrial disease published, but the group in Maastricht has at least a series that's higher than this. So there are about 24 papers with a mixture of stages. They have done cleavage stages. There's one paper in the Blastocyst with a baby boy that was born in a male-less mutation. And so they have done all this work, and let me show you how this is done. So this is a paper from Bert Smeets' group. Susanne Salleveld published this in 2013. And so here on the horizontal axis, you see the different embryos, and the mutation load is provided on the vertical axis. So when you look at this patient on the top, this patient had a mutation and was a carrier, and her embryos were occasionally biopsied several times. So if you have a blastomere that has a high mutation load, you want to, in those days, and we probably still want to make sure that that is the mutation load. And so you would take another blastomere out. And if the mutation load is high in both cases, then you want to test the assay, and then you take more blastomeres out because you're not going to transfer the embryo anyhow. And that's why you see multiple columns in these assessments. And so in this first cycle, unfortunately, this patient did not get pregnant because in all cases, you see that this patient didn't have an embryo transfer because in all cases, there was either no result or the blastomere showed a very high mutation rate. In cycle 2 of this same patient, there were only 3 day 3 embryos. And the last one, number 3, was transferred, embryo transfer ET. It was transferred. And so that's how this procedure is done. And I think it was carrier 4 who became pregnant, also from a second attempt, but there were embryo transfers in both cases. I presume this is a Lee syndrome, but I'm not entirely sure. I'm not very good in remembering these mutations. But Lee syndrome has these extreme levels, either you're affected very highly or affected not at all. And so all these embryos here were determined and had no mutation. And so in this case, embryo number 5 was transferred. And here, embryos 3 and 4 were transferred. And I think this second cycle resulted in a pregnancy, as far as I remember. So that's how that is done. And it's not done on a scale large enough to give you more data than what I just provided. But I think it is improving, and it should be considered before you do mitochondrial replacement therapy. And I hope that is done appropriately. I hear all sorts of reasons. Well, some patients they will not really have benefit from PGD. It never makes any sense to me. But I like to be convinced differently. And maybe those of you in the audience from the UK can explain this if we have some time later. Mitochondrial replacement therapy. There are 2 options, as you're aware of: spindle transfer, where you basically do this manipulation. This is a cartoon either from the New Scientist or from a popular journal, I don't remember. But you're replacing healthy with unhealthy mitochondria with healthy mitochondria. And you do that by nuclear transplantation. You have to visualize the metaphase spindle. And you take it out from the patient and put that in a spindle enucleated donor egg, reconstitute the egg that way, and then you fertilize it. The second option is nuclear transfer. Both options have been tried in animal models. The consensus among those who have a lot of experience say that this particular option is probably preferable, but we really don't know. There's I think not enough data in the human to argue that. But there you basically take the penuclei out and swap them between the donor and the patient. And so do you remove all the mitochondria this way? No, but if you do it very, very precisely, this carryover of affected mitochondria becomes very, very low, only really in the order of 1 or 2%, very low. If it's higher, then it's maybe technique-related. So there's wide support over the last few years, and this process started in the early 2000s in the UK, where they started debating this publicly with the HFVA involved. There's wide support for this. There was a public forum that lasted about 1.5 years, and the results of that were mostly positive. And so this has gone back to Parliament a few times, and now it's been approved, I think, since 2016. And the first licenses, I think, were given by the HFVA last year. And I think I don't know how many clinics, but I presume it's 1 plus. I don't know if that actually is publicly known. Yeah, it's only 1, okay. So there is a difference between, if you compare the United Kingdom and the United States, there's a tremendous difference there in the way that has been approached. In the United Kingdom, there has been this process protected and very long. I hope it's not going to apply to every little procedure that we're ever going to do in IVF, because patients will have no memory for this. They have no patience for this. So the licenses were given to do research again and again and again. There were intermediate research reports on this organized by the HFEA. Consultation was public, and then finally the green light was given and the license is out. In the United States, completely different. The FDA has argued it has jurisdiction. That started in June 2001, and that was a reaction to the popular press reaction to 1 or 2 papers we published on cytoplasmic transfer technology that was dating to the end of the 1990s, now about 20 years ago, and was the result of poor pregnancy rates and repeated implantation failure. And so the FDA reacted to that and said, well, that's okay, but you need a permit to do that. And so the permit process, they didn't go back to the House or the Senate in order to institute a law. They applied actually what the tools they had. So they applied a pharmaceutical drug protocol to the permitting process. So you have to write a product protocol system to apply that to embryos. Not the easiest thing to do. It's never been done, but it probably would have been possible. Certainly they were open-minded. People think that they prohibited it. No, they wanted you to go through that process. And so we actually applied, and then our group was separated, and the clinical and the research teams were in separate ways. And unfortunately, we stopped the application. Nothing to do with the application process itself. But we found the FDA very cooperative. That changed in 2016. In 2016, the FDA was told by a new law that had nothing to do with reproductive medicine, a law that was needed to pass the annual budget for 2017. They were basically said the FDA cannot get funding for any process related to germline genetic modification. Doesn't matter what it is or for what reason it is. And so the FDA they will receive and acknowledge that they have received your application, but then they will return and say we can't do anything without it because we're not allowed to look at it. It's an astonishing process. John Zhang, of course, was the first to have a baby for mitochondrial replacement therapy. This publication is from 2017. I think the baby is from 2016. It's a baby boy in a Lee syndrome case. This was done partially in New York City, and then the embryo transfer was done in Mexico. I guess you're aware of that. This paper is in Reproductive Biomedicine Online. The editorial is longer than the paper. The editorial is longer because there were so many issues with the process of submission and the process of permitting and the ethics involved and the consenting process, that the editors wrote this lengthy article about this. So I advise you, if you're interested, to read that. Okay, so what about safety? So is that technology safe? Well, we know very little about it. In animal models, it seemed to be doable, but most of these animal models were done in inbred species of the mouse. And the problem with inbred species is that you mate brothers and sisters for 30 generations, which is not equivalent to the genetic process in the human. And so you then get very awkward. If you start combining a nuclear transplantation, you get very awkward results. So it's very, very hard to do that and come to any conclusion. But the few that were not inbred shows that this was probably doable. And of course, several monkeys were born from this process, and they seem to be okay. We published a limited survey-based uncontrolled follow-up 2 years ago for our cytoplasmic transfer work. I know it's not nuclear transplantation, but you're mixing 2 populations of mitochondria. And so it would be interesting. We thought it was interesting to look at it. Remember, the groups and all the doctors had left from the days that cytoplasmic transfer was done. And so it was a little difficult to organize. So it was only a survey system that was filled in by the parents. But 12 of the 13 initial couples who had a pregnancy and babies born participated. And they received detailed questions about disclosure to their children, pregnancy, birth, health, and academic performance. Academic performance one was good, the others were all excellent. It sounds like cytoplasmic transfer is the way to go for reproduction. That was of course not what we intended to say. Of course, this is the result of environment. These patients were pioneers, and that means they were special. And so they were certainly not going to be not special when they were raising their children. So these children were in a particular environment, and that I think was the result of their very high grades in school. So they all did well there. The 12 out of 13 had normal BMI, which is in the normal limits of BMI in that age group. So these were all teenagers when they were tested, from 13 to 18. No current developmental disorders. Initially, and you can find this in Wikipedia even, you'll see there were 3 babies born with anomalies. Well, I don't know exactly where that comes from. What we did is report potential problems we noticed in early pregnancy. And one was the potential of an abnormal autism test that was seen in an 18-month-year-old. That patient refused to have further tests because the baby became a toddler and was developing quite well. And did well in school and is one of his excellent grades. So that particular child does not have autism, does not have spectrum disorder in the autism range. But one child has attention deficit disorder, so 1 out of 17 is kind of 1 out of 13 is kind of normal in the United States. So we didn't see any developmental disorders. The only interesting thing that came out of the study was a very low disclosure rate that this procedure was done to their children. So the parents were not inclined to tell them that they had actination, that they had IVF, and certainly not inclined to tell that they had cytoplasmic transfer. And we have no good reasons for that, except to say that there probably was a response to the very negative press in the years that cytoplasmic transfer publications were published, particularly in the last publication where we were showing the heteroplasmy in 1 or 2 babies that were born, meaning there were mitochondria from the donor and mitochondria from the patient. So the response in the press was so bad that these patients, of course, they read and they watch television, so they knew. And that may have been the reason for the very low disclosure rate. So what you could do is also selective elimination of mitochondrial mutations. I should have shown you the slide earlier. This was done in the mouse, not in mutations, but they looked at 2 inbred populations of mice. They were able to do mitochondrial DNA fingerprinting of these inbred populations. I have only 1 minute more, and eliminate with TALENs, which is the technology that was before CRISPR. With TALENs, they eliminated 1 population of mitochondria, and these embryos survived. They were transferred, and they gave healthy baby mice. And so, if you can do it with TALENs, you can definitely do it with CRISPR. CRISPR is much easier to replicate. That's why it's so famous. TALENs is complicated. This was a lot of work over many years and a lot of authors. So that has potential to do disease treatment in the human. Now there is work done with infertility, but because I'm now already 29 minutes up here, I think I'll stop here and just go quickly to my conclusions. So PGD for mitochondrial disease should be the first option for treatment, I believe, before MRT with nuclear transplantation. There's now 1 birth with MRT. Hopefully we'll get more, so most of the aspects are really unknown. We don't really know much at this point. In terms of infertility treatment and mitochondrial replacement or mitochondria transfer, there have been births reported sporadically in posters, 1 or 2 papers that are not in journals that we normally read. There's never been a randomized clinical trial. Augment is this process where you take embryonic stem cells or stem cell-like cells from patients from ovarian biopsies in a procedure that many people have doubts about. I don't have necessarily doubts about it, but I hear there's a lot of comments about that. And then the augment process, which hasn't been really implemented in good animal models. So there are many unknowns there. Then for infertility, where you actually can do the same as you do for treatment, for disease treatment, you can actually do nuclear transplantation and swap out the spindle or the pronuclei. There have been 3 births reported in the Nadia clinic in the Ukraine. No publications so far. They have tried to publish but have been unsuccessful. But they are going to present at ASHRE. They'll have a presented abstract, and that was accepted for presentation. So we're looking forward to that. And with this, I'm sorry I went a minute over time. Thank you very much.
Okay, thank you. Thank you very much.